Hello everyone, this is Dr. Anderson, and this will be a video presentation on the post-absorptive state. You know, we start with learning objectives, so we need to talk about some processes that happen in the liver. Glycogenolysis, or the breakdown of glycogen, and gluconeogenesis, the synthesis of new glucose from non-carbohydrate sources. These happen in the liver for the purpose of maintaining the blood glucose level that's important for maintaining brain function. So if the brain needs glucose, then the rest of the body is going to participate in a process called glucose sparing. Essentially, with glucose sparing, the rest of the body runs on fats and ketones so that glucose is spared for the brain. So what's going on with fat metabolism? We have to break down the triglyceride, that's lipolysis. We have to take the free fatty acids and break them down into groups of 2. And we'll get acyl units, 2 carbons that can either go into the Krebs cycle or be turned into ketones. The major hormone that dictates the reactions of the post-absorptive state is glucagon. Glucagon's main target is the liver, and essentially will be causing glycogen breakdown, gluconeogenesis, and ketone synthesis from the liver. Now we need some other hormones for the post-absorptive state. We need epinephrine so that we can harvest our fat stores. Epinephrine is going to be important for lipolysis. And growth hormone and glucagon also work together to maintain blood glucose level, as well as cortisol and glucagon. We're going to finish talking a little bit about calorie balance and the leptin hypothesis, or the lipostatic, essentially, hypothesis of regulating body size. So last time we were talking about the absorptive state. You'd just eaten a balanced meal of carbohydrates, proteins, and fats. Well, with the post-absorptive state, it's been 4 or more hours since food has been ingested. And so instead of storing excess glucose, you need to maintain the blood glucose level, especially for the metabolism of the brain. The brain is just not able to work on fat. They can use ketones, but lipid metabolism is not favorable in the brain. So we need to break down the molecules that we stored in the absorptive state so that we continue to meet the needs of the body even though food hasn't been ingested for several hours. So where are we going to get glucose? A major place we get glucose is glycogenolysis, or glycogen breakdown. This happens in the liver. The liver breaks down glycogen to increase blood glucose level. And the reason it can do that is that the liver has the enzyme that's able to remove the final phosphate group so glucose can leave the liver. Skeletal muscle is a tissue that stores glycogen and breaks down glycogen, but skeletal muscle cannot remove that final phosphate. So glucose, the phosphorylated form of glucose, must enter glycolysis. Now, pyruvate or lactate resulting from glycolysis, the pyruvate or lactate could be put in the blood and sent back to the liver or sent to other tissues. But the skeletal muscle cannot raise blood glucose level. The other process happening in the liver is gluconeogenesis. So gluco as in sugar, neo as in new, and genesis as in creation. This is the synthesis of glucose by the liver from non-carbohydrate sources. And examples are glycerol, some amino acids after they've been deaminated, pyruvate, and lactate. What do these amino acids have in common? What do these compounds have in common? They all have 3 carbons. 2 3-carbon molecules can be put together to form a 6-carbon glucose. So here we are in the liver. Glycogen is going to be broken down to form glucose. Gluconeogenesis is going to be performed. So glucose is going to build up within the hepatocyte. Without insulin, These processes occur. Glucose builds up inside the cell, and there's a gradient for glucose to leave. Remember, during the absorptive state, insulin was occupying this site, and that promoted glucose coming into the cell, being phosphorylated and stored as glycogen. Here, we're dephosphorylating glucose and allowing it to leave to maintain blood glucose level. So with glucose sparing, we're making some glucose, but the priority for that glucose is the brain. Neural tissues really prefer glucose to make ATP. They can run on ketones, but having them run performing beta oxidation and lipolysis is not going to work in neurons. So the other issue is that we're saving glucose for the brain because during the post-absorptive state we simply cannot make enough glucose for everyone. So other cells have to switch to another substrate, and that substrate is fat. So as cells switch to fat metabolism, glucose is then spared for the brain. So adipose and resting muscle, notice there are no GLUT transporters in the membrane. Insulin is not bound to its receptor. Glucose transporters, the GLUT transporters, are in the cell, in vesicles, and if the transporters are inside the cell and not in the plasma membrane, then glucose has no way to enter, and these cells have to convert to fat metabolism. So lipolysis is a breakdown of a triglyceride, which is composed of one glycerol and 3 fatty acids. These are covalent bonds, and so you can use lipases to break down these bonds, and you have a 3-carbon glycerol that can be sent to the liver for gluconeogenesis. But fatty acids cannot be used to make glucose. They're broken down into groups of 2 by beta oxidation. That permits cells to convert those acyl groups into acetyl-CoA, put into Krebs cycle, make ATP. It's a wonderful, wonderful source of ATP. But 2 carbon molecules cannot be used to make glucose. So free fatty acids are not gluconeogenic. They are ketogenic, but not gluconeogenic. So here are lipases working on triglycerides. We have a fatty acid. They're broken down into groups of 2 by beta oxidation. We have acyl groups that can enter Krebs cycle. They become acetyl-CoA and are churned through the citric acid cycle to promote ATP synthesis. The glycerol from the triglyceride can be converted back to glucose, and then glucose can be burned through glycolysis. Then the pyruvate that results can be put into the Krebs cycle. So what is a ketone? When fat is broken down faster than it can be burned in the Krebs cycle, the acetyl-CoA groups can build up and they react and they form a ketone or a ketone body. Examples of ketones are acetone, acetoacetic acid, and beta hydroxybutyric acid. So these are all combinations of acyl groups. So the liver can make ketones with the help of glucagon. Those ketones can be put into the blood and sent throughout the body to be used for energy. And of course, that saves glucose for the brain. Please note that some parts of the brain can use ketones. So those ketones being sent out by the liver, some of that also fuels the central nervous system. So in the fasted state or the post-absorptive state, glucagon is the dominant molecule, dominant hormone. So as glucagon secretion increases, it has its effects on the liver to increase glycogenolysis, the breakdown of glycogen, gluconeogenesis, the creation of glucose from non-carbohydrate sources, and ketogenesis, the production of ketones from the products of beta oxidation. So glucagon responds to hypoglycemia. When your plasma glucose goes down, alpha cells in the pancreas are aware of that. They are also sensitive to high levels of amino acids. High levels of amino acids also stimulate insulin. You might say, well, what the heck is going on there? Well, if you have amino acids and glucose together, you want to stimulate insulin. However, if you have amino acids and no glucose, you want to stimulate glucagon so you can raise your blood glucose level while you're using the amino acids. So the alpha cells are in the pancreas, they secrete glucagon, glucagon goes into the blood. Please notice that glucagon's main target is the liver, and the liver will break down glycogen to produce glucose. The liver will use lactate, pyruvate, some amino acids, and glycerol to make new glucose. This raises blood glucose level. Fatty acids are sent to the liver, made into ketones, and ketones can be used by the tissues to make ATP. So notice it says prolonged hypoglycemia. So when your blood glucose level goes down for a little bit, for a little while, simply the removal of insulin helps you progress into the post-absorptive state. But if the hypoglycemia is low enough and long enough, then glucagon is secreted to promote even more post-absorptive reactions. So here's a little table on glucagon coming from the alpha cells. It's a peptide made out of amino acids. It's water soluble. It's dissolved in the plasma and does not need a carrier. Therefore, it has a very short half-life, 4 to 6 minutes. And when your blood glucose level goes down well below 65 milligrams per deciliter, or you have amino acids in the blood, that stimulates glucagon. The target cells of glucagon is really the liver, binds to a glucagon receptor. You activate a G protein coupled receptor that increases intracellular levels of cyclic AMP, so it's working through a second messenger system, and then cyclic AMP turns on the kinds of enzymes we need. Enzymes that will break down glycogen, enzymes that will produce new glucose, enzymes that will create ketones. So it's going to turn on enzymes fast. It's going to have some long-term effects on the kind of enzymes you make. If your blood glucose level is too low, you're going to do things to raise blood glucose level. When your blood glucose level comes back up, then you're going to turn off glucagon. So beautiful negative feedback mechanisms going on here. So this is a complicated diagram showing you how your liver, your adipose tissue, your muscle, and your brain are all interacting during the post-absorptive state. So initially, your liver is breaking down glycogen. Glycogen becomes glucose to supply the energy needs of certain tissues. Free fatty acids undergo beta oxidation. The liver makes ketones to fuel the body. Adipose tissue is worked upon by epinephrine. Glucagon doesn't really affect adipose tissue. So epinephrine, it's stressful to have low blood glucose levels. So you produce epinephrine. Epinephrine binds to a beta receptor that increases lipolysis. Free fatty acids are sent to the liver for ketone synthesis. Glycerol is sent to the sent to the liver for gluconeogenesis. Fatty acids can be sent to the muscle to fuel its needs. Some proteins are broken down, and the amino acids are sent to the liver for gluconeogenesis. Muscle can break down glycogen. Glucose 6-phosphate that comes from glycogen breakdown is shuttled into glycolysis. A byproduct of glycolysis is pyruvate or lactate. Both of these molecules can be sent to the liver in the service of gluconeogenesis. Glucose produced by the liver fuels the brain. And note that some parts of the brain can also use ketones. So what about other hormones, given that glucagon works on the liver? How do we get effects on muscle and fat? Well, we're relying on epinephrine. Epinephrine increases fat breakdown. Growth hormone and cortisol, remember, are permissive for glucagon, and that's going to help get our blood glucose up. And simply having less insulin also is going to promote the reactions of the post-absorptive state, like fat breakdown and lipolysis. So this is a summary slide. It comes from a different textbook. This is what we did in our previous lecture, showing our storing catabolic reactions, showing that most cells are running on glucose, and the liver is storing extra glucose as glycogen and fat. This particular presentation, we have been harvesting our stores, performing catabolic reactions, amino acids from proteins, glycerol and fatty acids from triglyceride, glucose from glycogen. Most cells of the body are running on fatty acids and ketones, which spares glucose for the brain. The liver is helping to maintain blood glucose level by taking pyruvate, lactate, glycerol, and some amino acids and turning them into glucose through a process called gluconeogenesis. We're also breaking down stored glycogen in a process called glycogenolysis. So in summary, the post-absorptive state occurs when we're beyond 4 hours after a meal. Catabolism predominates. We are breaking Okay, my presentation is still running. I beg your pardon for this technical difficulty. Oh, Lord. Okay, let's try that again. So we're beyond 4 hours of having a balanced meal. Catabolism predominates. We're breaking down the things that we stored earlier. Most cells are using fatty acids and ketones so that glucose can be spared for the brain. The liver is breaking down glycogen to raise the blood glucose level and synthesizing new glucose from lactate, pyruvate, glycerol, and some amino acids. Therefore, these are molecules that are gluconeogenic. Fatty acids are ketogenic. They are not gluconeogenic. So, a little bit about metabolism and weight. Eating behavior is very complex. People eat for a lot of reasons besides being hungry. They eat because it's a holiday. They eat because they're bored. They eat because they're nervous. They eat because they're watching football and a commercial comes on for pizza and then they think they want to have pizza. So there are lots of complicated things. And there's lots of arguments about how metabolism works, but calories eaten versus calories burned must balance out. So why is it that a person who's struggling with weight and maybe doesn't seem to eat very much has trouble losing weight? And it's because their metabolism is perhaps lower than it needs to be. So we have to think about both diet and exercise. And maybe thinking about hormone imbalances as we approach the problem of obesity. So I want to tell you about a couple of theories. There's one called the glucostatic theory, that glucose metabolism by your hypothalamic centers regulates your food intake. So if your blood glucose level is low, you're hungry and you want to eat. If your blood glucose level is high, you're not hungry and you don't want to eat. Of course, people are interested in things like the glycemic index of foods. If you eat a bunch of sugar, that may decrease your appetite, but then you stimulate a bunch of insulin. Insulin gets the glucose into your cells, your blood glucose level goes back down again, and then you're hungry again. So, that theory doesn't explain everything. We also have a lipostatic theory, and that is your adipose tissue is an endocrine gland, and it produces all sorts of hormone signals that regulate your metabolism and your eating behavior. So leptin, neuropeptin. Y, I just realized I have a typo. This should be ghrelin. CCK, CCK is a satiety factor. GLP, orexins, these are all interesting peptides. I'm going to take a moment to say something about leptin. So our fat stores produce a molecule called leptin. So if you gain weight and you increase your fat stores, you increase your leptin secretion. Leptin inhibits something called neuropeptide Y, and through other mechanisms has an effect on the hypothalamus. And essentially what happens is as you increase leptin, you decrease your appetite and you increase your metabolism. That changes the energy balance and gets you to lose weight or lose fat, so you're back to your fat set point or your adipose set point. Likewise, if you lose weight, if you lose fat, then leptin goes down. You feel hungry because you're not inhibiting neuropeptide Y. This makes you feel hungry. So you're feeling hungry and your metabolism goes down. So you end up gaining weight and getting back to your initial set point. So, so there are a lot of theories about why this goes astray. Do people who chronically overeat then downregulate this system, and so then it's not as sensitive? We have a lot of cultural norms about eating 3 meals a day or those kinds of things. We may want to revisit that so that people eat when they're hungry, and if they're not hungry and they don't want to eat, we shouldn't make them feel guilty about not wanting to eat. So there's a lot to be researched and a lot to be examined in how our bodies regulate our fat stores and regulate fat metabolism. But I really would like you to appreciate that your fat tissue, your adipose tissue, is an endocrine gland. It produces a hormone called leptin, and increased levels of leptin decrease your appetite and increase your metabolism, and it works through the hypothalamus. So it's been a wonderful semester. It's been a wonderful class. I hope you find this presentation helpful, and I wish you all the best. You've been wonderful students, and I thank you very much. So I'll see you next time.